polymerase will start copying the DNA sequence, and to give the copied DNA. Now we're interested here, so we're not interested in this strand anymore. We're interested in this strand. So when the DNA polymerase start to copy the DNA sequence, when it reach the probe, it will start to degrade the probe. Okay, so it will start to degrade the probe. Now, when the fluorophore is degraded from the probe, and so it's not close anymore to the Kencher, and so the Kencher cannot absorb the fluorescence coming of the fluorophore anymore, at this point, we can see the fluorescence coming out of the fluorophore, because the Kencher cannot inhibit the fluorophore anymore. The Kencher cannot absorb the light coming out of the fluorophore anymore. So the polymerase will continue by copying the DNA and by degrading the probe. Okay, like this, like as you are seeing here. So the probe will be completely degraded when the polymerase is copying the parent DNA. So let's go back here. So here, when the probe is degraded, we can see the fluorescence coming out of the fluorophore. Okay, then the polymer will continue copying the parent DNA. Now, how does this work? Now, when we have, going back to the normal PCR, when we have the PCR, then we will get more and more sequences at each cycle. So let's say we have TaqMan in the medium. So in each cycle, there will be more DNA sequences and more DNA strands, and then more probes are binding to the DNA, and more probes are degraded each time. So at each time, so at first, let's say we will get a little bit of fluorescence, then we will get more and more and more fluorescence, and this what can be detected by the spectrophotometer. We'll see spectrophotometer. We'll see in here. Now let's speak about a little bit about the advantages and disadvantages of the specific detectors, or the TaqMan. Of course, the first advantage is the specificity. It's specific. It's specific to a certain sequence or to a certain gene. And because of this, I can do the multiplex real-time PCR. What is the multiplex real-time PCR? It's to perform real-time PCR to many genes in one tube. And yeah, I can do this with the specific detector, because let's say we have gene 1 and gene 2. We can use two different specific detectors. The first is coupled with the blue fluorophore. The second is coupled with the red fluorophore. We can also use green fluorophore. We can use multiple specific detectors, or multiple TaqMans. And then this will be very specific to G1. This is specific to G2. And then if I have one DNA sample and I want to perform several real-time PCR in one tube, I can, for several genes, I can use the specific detector. Some of the disadvantages, it's hard to sequence because it needs to be highly specifically sequenced. So it's expensive. And there is a probability to false results, which is derived sometimes from the inability of the Kencher to totally inhibit the fluorescence coming out of the fluorophore. Sometimes the Kencher will be not so effective in inhibiting the fluorophore, which might give false results, but this is rare. There is very important application in the real-time PCR is the reverse transcription. It's not an application, actually it's not an application, it's reverse transcription is highly used with real-time PCR. So let's say we have RNA virus, or we have a human cell, and we need to measure the expression of a certain gene. We need to measure how much this gene is expressed. Why? Because some genes, the expression of some genes differs between normal cells and cancer cells, for example. So the measuring or the analysis of gene expression is highly used in cancer cells. In this case, we can in this case we take the messenger RNA sample out of the cell, or extract the messenger RNA extract out of the cell, and we undergo something called reverse transcription to produce the complementary DNA out of the messenger RNA. Now I'm going to speak really in general about reverse transcription. So in order to perform reverse transcription, we need the parent messenger RNA. We need also the primer. We need the DNA contents to produce DNA. We need a very specific enzyme called the reverse transcriptase, which is used for reverse transcription. We need also RNAs, and we need DNA polymerase. Now, how reverse transcription is done? It's like this. So we have messenger RNA or RNA sample. So the reverse transcriptase copy the messenger RNA. It copied the messenger RNA. It produced DNA from RNA. So it's not transcription. In the normal transcription, we copy DNA to RNA. In the reverse transcription, we go back. So we copy DNA from RNA. Okay, so we copy the messenger RNA, we get something we call the cyclic DNA. After this, the RNAs will degrade the messenger RNA, okay, and then the DNA polymerase will copy the first strand of DNA, and then I will get double-stranded DNA from the RNA. I can use this double-stranded DNA in the PCR, in the real-time PCR. This is highly applicable in real-time PCR, actually, as I told you, in cancer, in the diagnosis of HIV or other RNA viruses. It's highly applicable. Now you know that in real-time PCR, the spectrofluorimeter measures the amount of DNA in the sample continuously during the PCR process. But how can I get the data? Or yeah, the amount, how can I get the data? So the quantitative real-time PCR, of course, real-time PCR is quantitative. So this is the machine, let's say, of real-time PCR. Normally it's engaged with a computer program, and which gives me the data in this form. Like, this is what we call the amplification curve, or in this form, or in this form. Anyway, it's the same. We have the same elements. So this is the fluorescence. What we see here in the y-axis is the fluorescence. In the x-axis are the cycles of PCR. Here we have 38 cycles, for example, 40 cycles. It depends. And this is the amplification curve. So you're seeing multiple curves because, as I told you, we can apply real-time PCR for several samples at one time. We can apply it in the 96 Wells plates. And so we get several amplification curves. You are seeing here three overlapping curves, because sometimes we apply the same samples 3 or 4 times, like in a triplicate, to ensure about the results. So this is what you're seeing here, or this is what you're seeing here: multiple amplification curves overlapping over each other, because they are amplification curves for the same sample. So we apply the same sample in several tubes to ensure about the result. Now, in all the curves you're seeing here, we have general elements. And the elements are, so this is the fluorescence, as I told you, this is the cycles. And this is very important. It's called the threshold line, what you're seeing here, or here, or here, the threshold line. And this is the most important value because it's the CQ value or the CT value. It's the threshold cycle. It's called the threshold cycle. And the threshold cycle, it's the cycle at which the amplification curve intercept with the threshold line. So the intersection between the amplification curve and the threshold line. It's called the CQ or CT, the threshold cycle. Why it's so important? Because if you see here, going back to here, for example, the CT here is somehow 22 at cycle number 22, but here it's at cycle 31. So let's go to here. What you're seeing here is that the fluorescence coming out of the sample at first is too minimal, and then as PCR goes on, it starts to increase. So when we have higher amount of DNA at the beginning, the amplification curve will go faster because we have higher amount of DNA from the beginning. And when we have very little amount of DNA from the beginning, the amplification curve will start to increase in a delay. So the curve which cross the threshold line first is the sample which had the higher amount of DNA at the beginning of the PCR, and the sample which crossed the line lately is the sample which had the less or the least amount of DNA at the beginning of the PCR. So we use this to compare between samples. So if I have several samples, I can use real-time PCR. Then I can know from the CT which sample had the highest amount of DNA at the beginning. So this is very important value, the CQ or the CT, and you can see here after several or certain cycles, the curve becomes non-exponential. Here we have the plateau phase because after several cycles we don't get any increase in the amount of DNA anymore. Maybe because the polymerase become inactive after a certain time, or because because the polymerase becomes inactive after certain time. And here is the exponential phase in which the amount of DNA increases exponentially. So this is everything I wanted to tell you about real-time PCR. Yeah, if you enjoyed the video, don't forget to like, don't forget to share it with your friends. And if you have any questions or any question regarding real-time PCR, don't forget to leave your question in the comments because I will answer you. If you enjoy my videos, don't forget to subscribe my channel. There will be many other interesting videos. So yeah, so subscribe my channel, like the video, leave your comments, and see you in the next video. Bye.